You want to, you're driving, that's a downforce. So
Okay.
So you want less
Lower.
downforce if you want to float up, right?
Gotcha.
So for anyone listening on the web, what's being described is we took on weight with that last rock. So the Z bias, please jump in if I get this wrong, is effectively the trim for how buoyant Hercules is.
Yep. So it's adjusting the vertical thruster to how much it's thrusting down to, you want to be like neutral, so you're not going up or down.
You're just dealing with the topography, the slope.
Yeah.
Go up when you have to.
When it's convenient, we'd like to get a Niskin sample from up
Okay.
here.
Do you want to hold position for that?
Not necessary.
Is it necessary for you?
If it's necessary for you.
Not necessary for me.
Okay.
Okay.
All right, I'm swapping out with
How
Rennie.
close to the bottom?
Just right where you're at. Just hold it there.
Okay.
Just watch your altitude. Just keep it steady.
Okay.
And then I will.
Science shift
do my
change. Thanks,
thing.
everybody.
Thank you.
You want to zoom out?
Can we rack the camera back?
Yep. Camera back.
Pan over to the left a little. And this is Niskin number 2?
Yeah, but hold on. We don't have view of it. Okay. I'm watching.
What sample number? Kayla, we're taking a Niskin. Could I have a sample number?
Are you ready?
Yep. Good.
Okay, so Niskin 101.
I think I parked it.
Wow.
I had to go from above. Okay.
Are we? Okay.
This will be Niskin sample NA124 101.
I think so.
Bridge now.
I have it.
Hold position here.
Just doing a
Thank you.
bit of a shift change here. Our back and front rows are getting oriented.
Now, could you pull up the last target for me on the map just so I can copy that down for the Niskin? Thank you so much.
Copy that. Actually, it was not dropped, I don't believe. Let's see. This one was. 100 was the rock sample. So this one will be the Niskin, and that was 101.
Yes.
Oops. And do we know what Niskin number that was? Was that number
Niskin
2?
number 2, yes.
Thank you. And 473. Did you need that target still up, or are you good?
Yes, please. I just got to copy that down.
Sure.
Sorry.
No worries.
Got it. Thank you.
Sure.
Hello, folks.
Hello, hello.
Hello, let me just zoom out here on the map, see where we are.
Good afternoon, everyone. We've had a shift change, and we now have Jonathan on video, Jake piloting the Argus, Jessica piloting Hercules, and Renee doing nav for us.
Hello.
And on the science crew, we've got Kyla on data, Caitlin on data, Paul running the watch, Paul Jensen, and Grey Grouse here. We're sitting on the Cortez Bank, a famous surfing place west of San Diego in Southern California. Also well known for scuba diving on top, but we're sitting at about 470 meters on this rubble field, and we'll continue going a little bit shallower. We're pretty much in a very flat area, but we're exploring for different kinds of rocks and the animals and microbes associated with them.
Roger. So, I think are we all clear to just continue on our eastward move there?
Yes, please, Jessica.
Roger that.
095.
095, Rog.
Bridge, nav. We'll have a ship move 50 meters, bearing 095 and speed 0.3. Thank you. Apparently there's a lot of siphonophores here.
Yeah, that was a big siphonophore.
Yeah.
There's one also on bubble there, isn't it? Kind of captured on bubble.
Yeah, I saw one
Yeah.
when it was out facing a different way.
It's taking a ride. Hitchhiker.
We're seeing a lot of carbonate rocks here that are pretty flat, but then the occasional black rocks as well, which are more of interest to us, but they're all covered with a lot of different sponges. The bushy white things are a sessile sea cucumber. Some big carbonate slabs. We'll be just going along a little bit before we settle down and try and pick up a rock or two.
Roger that. And are you guys interested in transects along the way as well?
Sorry, Jessica, missed that.
Oh, are you guys interested in transects along the way as well?
Oh, yes, we will.
Roger that.
When was the last transect, actually?
The end of of transect 2, which I believe was the last one, was
45 minutes ago. Oh, quite a while ago.
200 meters away.
Yeah, we could do a transect now while we get oriented.
Sure thing.
Thank you.
Yeah. So this is going to be transect number 3.
Copy.
Rog.
Quick gauge checks.
Roger that.
Ready when you are.
All right, ready. Ready, ready. stepping forward.
All right, let's start there, Data.
Oh, okay, copy, thank you.
Sounds good, thank you.
So we're doing a transect here along the bottom, a few meters up, trying to stay, giving us a similar field of view. The lasers you can see there, the green, are 10 centimeters apart, and this video transect of 100 meters will be combined with others from all the various sites and quantified to give us an idea of the distribution and abundance of large animals at different depths and different oxygen concentrations along the various sites we've explored during this cruise.
We were catching up today on some of the press releases that have come out on this cruise. Really quite surprised to see how much publicity there's been. Scripps release had a press release before we left. And just saw earlier today an article came out in the San Diego newspaper. Another article came out in Wired magazine. It's possible that
Oh, I don't think we can hear you anymore, Paul.
Yeah, I was adjusting my microphone, sorry.
All right.
That
That's great.
delicate space between
Much better.
where you can hear or
Millimeters.
background noise, yeah. Yeah, so I was just talking about the public interest that there is in this cruise, and I think a lot of that reflects the fact that there's so much in the ocean that's even close to home that we really don't have an understanding of. And I think the idea that there's what we're calling biopharmaceutical potential in these deep habitats and bringing awareness to that, which I think helps encourage us and hopefully decision makers to think carefully about how these habitats are preserved. So even though we're really right in our own backyard, there's a lot out here that hasn't been explored. And one of the things we hope to do as part of these explorations is to better understand if there are microbes and various animals that are producing molecules that we would say have biopharmaceutical potential. So that brings a new level of interest, I think, to the value of these habitats, besides their intrinsic value to just supporting the community and ecosystem function out here.
Big Siphonophore.
So we were watching from the lounge, and I think that yellow sponge we're passing now was collected. And I think there was at least one other sponge collected.
A big white one. And we keep seeing a bunch of these along these transects. These yellow ones look a little bit different from the previous day's dives. So we're really interested in getting an idea of all the different sponges. And it's really difficult to identify a sponge just by looking at it. Video usually takes a lot of hard work to identify these. So you might hear us refer to them, the yellow sponge, the pale yellow sponge, and they could all be very different or very similar. It definitely looks like we've got some different sponges at this site today.
Yeah, now we can use molecular techniques to identify animals. But what's also traditionally used for sponges, they have these spicules. And the composition of those spicules and also the shape of those spicules is very diagnostic of who they are. So oftentimes it takes an expert in evaluating those spicules to classify the sponges. And in the old days, at least, you would send your sponge sample to someone who was really good at doing these identifications. But now with molecular techniques, we can sequence genes, and this is something that Greg Rouse specializes in, resolving the relationships of these organisms based on DNA sequence data, which in many cases tells us that things that we once thought were the same groups are actually different groups. And some things that were put in different groups actually should be in the same group. And there are a lot of challenges to deconvoluting all of these relationships and trying to get organisms into their right grouping so we can assign names to them, meaningful names to them, and then communicate with each other and make sure we're all talking about the same organism when we're speaking.
Yeah, that'll be especially important if they find some particular sponge here that we've called yellow sponge, for instance, on dive 1842. If we want to come back and get that sponge, we'll need to be able to identify it. So we're putting pieces into the permanent collection at Scripps, as well as we'll sequence some. So we've got a good reference to hunt these sponges down later if they turn out to be of significance for pharmaceuticals. That is an interesting looking rock.
Yeah.
Is that a sea whip we're coming up to?
Can't hear you, Kelly.
Oh, a sea whip?
Yeah. Yeah, we're
A
starting
couple
to
of these
see a few of
popping up.
those.
Yeah,
Yeah, that's
a little bit.
probably a primnoid that we might have gotten yesterday.
See a couple of them on some of the rocks.
One of the interesting things that we were looking at last night was a map of the topology of this Cortez Bank. And it was surprising how, well, somewhat surprising, much of the deep sea has yet to be mapped. The vast majority of it, in fact. But we were a little surprised that just off San Diego, there was no really good map of this Cortez Bank. And so after we're doing our dives during the day, at night, there's always mapping activity going on. So we have dedicated staff here, Lindsay and Aaron, who work on doing the mapping. And they made a great map of part of the Cortez Bank last night, which helped us choose this dive track. And this requires a ship to go backwards and forwards in a zigzag pattern, sending down It's called side scan sonar to give a high resolution map of the seafloor.
Multi-beam sonar, actually.
Multi-beam,
Yes.
not side scan. And what's the resolution on the sonar?
It depends on the depth. In an area like this, I think probably Lindsay was able to grid it maybe 10 meters, something like that. 10-meter grid cell size.
How many meters? Sorry, I missed that.
Probably 10. I think if you
10
did
meters.
several passes in different directions and kind of you get it down a bit lower than that, but you know, finer. But
And where do you deposit the mapping data?
so we record it all in our own archives, which then gets sent to R2R, which is Rolling Deck 2 Repository.
Right.
And from there, NCEI, National Center for Environmental Information, grabs that data and puts it in kind of a public repository. So it's all available publicly within a somewhat timely manner for how many hands it has to pass through. So first and foremost, it's available by request through us if it's not yet up on those archives.
Yeah, there's been a big push to take advantage of these data sets and make them available to the public, I know.
That's the end of transect right there.
Beautiful,
Thanks,
thank you.
roger
Just
that.
a couple meters over, sorry about that.
No
So
worries.
could we slow up a little bit and
Sure thing.
we'll look at this feature on the left here.
Yeah,
Bridge
let's
nav.
do it.
This looks really cool.
Let's hold position here.
How's that?
Hagfish at the base of that.
You want to
Gorgeous
there?
Brisingid.
Wow.
Do a partial zoom there, Jonathan.
Got it?
Yeah, that's great. See what's living along the crust.
Oh, look at that hagfish.
We
Kinda
do love
hide.
our hagfish.
Yes. Oh, it's kind of curling around there. Hello.
Oh.
Hey,
Oh, very cute.
scratching its back.
Getting rid of some of that extra slime.
So hagfish have this absolutely fantastic mechanism of being able to latch onto whatever meat they're eating, say a dead whale, and they tie their bodies into a knot, and that knot moves from their tail to their head, so they're able to have enough pull to take that nice chunk of meat off the whale.
Awesome.
Which they need to do because they don't have a lower jaw.
Mm.
They just have a jaw with an upper
Right there,
jaw with
Jonathan,
teeth
please.
on it. If we could settle down here, please, in front of the flange there, and
Sure thing.
anywhere you want, please, Jessica. And we might try a slurp on top of that flange.
Slurp on top of the flange,
Yes.
Raj. Around here will probably get us the best reach. Would that be all right with you?
Yes.
Great.
Thank you.
See grass down there. All right. So I've been trying to come up with some more deep sea jokes, and at the moment I'm kind of falling short. So stay tuned for those. However, I do have an ocean themed joke if you guys want to hear it.
Absolutely.
Okay. So what do you call a seagull that flies over the bay?
A bagel.
Yes.
I love it.
Yep.
With cream cheese.
And what is a sea lion's favorite class that it took in high school?
Ooh. That took in high school?
Art.
Uh,
That's cute.
Wow.
Where's the hockey puck? Huh. They left us no viz here, did they?
No,
Oh, all right. Just have to go and see what we got.
a little bit in bubble.
Yeah.
So this gives us a good sense of why we can't open that drawer any longer. If there's a rock sticking out.
That rock is blocking.
I'll give you a little iris. I think you'll see the.
Sure. Oh, that helps actually. Okay, we're gonna keep this trick for next time.
There it is.
So I'm not up on.
So what were they thinking on watch 1 with that rock and getting it into that bio box? I don't know.
They're trying to stack everything on there.
Stand by.
Ooh, ooh.
Come on, we did a slurp sample with it on there.
All right, let's try that grip out. Yeah, go ahead and give it a flush.
What do we got?
Yeah, where are we gonna go with this guy? Now that we got a precarious grip on it.
Finer course is the
Can
question.
we look at the top of the flange there a little bit? Just
Yeah.
scan around, please,
Sure thing.
Jonathan.
Go ahead and push on in there, please, Jonathan. That's great.
You could go in a bit closer. We'll just see how coarse the sediment is.
Sure thing. Got any more zoom on there, Jonathan?
We got
So
zoom. That's
there's
back.
a lot of tubes. Yeah, if you could slope over that kind of area, please,
Sure thing.
that would be great.
Would you like this in fine or coarse, Greg?
Fine, please.
Fine. So we could go for jar number 2.
Jar number 2.
Yeah, come wide there, please, Jonathan.
Oh, went the wrong way. Jar number 2.
Roger that. All right, Jonathan, go ahead and push on in. Wonderful. Wonderful.
Wunderbar.
Oop, too far. All right, go ahead and start some suction. This
Coming
area
up.
all right for you guys?
Yeah, this looks good.
Roger that.
Yeah, if you can knock those tubes over, that would be,
These
hopefully
ones-
they'll come up. Not sure what they are.
Yeah.
Maybe
Keep
old
it
sea
rigid.
whips.
Oh, putting up a fight.
Oh yeah,
Got one.
great.
Nice.
Might avoid the long one in case that gives us a clog, but I'll go for the smaller
Yeah.
ones over here.
No problem.
What would a Sunday be without a little vacuum cleaning?
Oh, yeah. Oh, today is Sunday. You are right. Very nice. Very good. Sundays on the ships, we always have ice cream. So.
Ice cream day.
Ooh.
Yeah.
Something to look forward to.
Ice cream and
Good
potatoes.
to have that advanced knowledge so we can save a little room.
Oh. This tube does not want to go.
Yeah, he's stuck.
Very, very long.
And push in a bit there, please, Jonathan. There's someone that's branching out next to him, though. This guy here. Oh, he's on a little rock. He might not come in very easily.
I'm not sure what that is.
Get in there. Hey.
Oh, you got it.
Got it.
Thank you.
Amazing.
Boy, there's a little escaping
Wonderful.
worm.
Oh,
There's a little
Yeah,
scale
there's
worm.
a worm.
Ooh.
So it's no longer there, so it's probably
Great.
in there.
It's going
Not
to be
many-
hard to see him in that vacuum.
Surprised we don't have more stuff in there.
Rock didn't come through yet.
Might be against the mesh.
What about the little puff sponges?
Yeah, what else do you guys want here?
There is that tube with its mouth up towards you. Do you think that's, is that?
Yeah, this guy here?
Yeah.
Yeah, let's try. Oh, you are a long puppy. Sorry, 1 second. Come wide a little bit, please, Jonathan.
I know.
I'm gonna reset my grip. It's not the best grips, but. Okay, go ahead and push on in there again, please. He is pretty stiff in there, isn't he?
Oh.
Come a little wide. Okay, go ahead and push back in there, please. Oh my goodness.
Oh, that's a long tube.
Come wide, please. Like I'm caught. And a 0 suction, please.
0.
Rog. All right.
Yeah, I think they were feather duster worm tubes, but
That's
they're
full
super
wide.
leathery and the animal's probably going down into the rock.
Yeah. Should have reset myself a while ago. Go ahead and push on in there a bit, please, Jonathan. That's great.
Just confirming this is sample NA124-102. That went to jar 2, slurp jar number 2.
Copy that.
All right, this is a much better grip for us to do better work. Go ahead and push on in there a bit. That's great. You guys want me to still go for this tube here, or do you want some more
Yes,
up
please.
here?
Give it another try. And
Roger
the little tuna crab up to the left looking out at you could be the next target.
Roger that. Okay, go ahead there, Jake. Come a little wide, please. This one has become a nemesis.
Stubborn.
Stubborn indeed.
Yeah. Need like a little scrapey flat bit on the bottom.
All right, let's go for the tuna crab real quick, and then
It's worm 1,
For now,
Herc 0.
till we hit it.
We might have one in there anyway.
Go ahead and push on in again, please. Actually, come a little wide. A little wider. Thank you. Jake, would you mind looking at the base of the hose, please, and seeing if we're at the red tape? That is occluded by my arm. Full wide, please. I think that squat lobster is just right outside of
Just about at the yellow tape.
Reg. Let him push on in there again, please. I think we might be able to reach it if it doesn't move. No, negative. It's just outside of our reach.
Okay, no problem.
Come a little wide. We can look somewhere closer
Yeah.
and somewhere easier to grab.
Can we pan right? Maybe under where the arm is, there's like a white granular sediment.
Roger that. Full wide there, Jonathan. Over here. Go ahead and push on in, please. That's great. Anywhere around here?
Yeah, just past the brittle star.
Just past the brittle star.
Oh.
So
Away from us. Yeah, if you can reach that far, further past. Yeah, that's it.
right here. Roger that. Go ahead,
Perfect.
Jake. This surface layer here.
Yeah, it's all tubes. Some of it might come off. It's pretty hard rock, not a lot of sediment. Now I see, if you come left, Jonathan, can you zoom in straight
Go
to
ahead, Jonathan.
this part? Yeah.
All right.
See the worm, it's got they make these tubes that put out little fingers.
Yeah, this one here.
Yeah, and they coat them with mucus and that traps plankton and then they come out and eat it.
Roger that.
So that's a kind of spaghetti worm.
Wow.
What are those little brown ones that look like pills?
Little Tic Tacs. Weird.
Yeah, we saw some of those earlier and we were wondering what those were.
Grommets?
Grommets?
Grommet?
They're the little egg-shaped things that are attached to the rock.
See another one of them here.
Wow. Look at that.
Jake, are you full section right now?
Yes.
Okay.
Sounds like these two worms have convergent evolution with spiders, building a net-like structure and capturing its prey.
Yeah, and worms do it, a few different groups of worms do it that are normally stuck in tubes. They've evolved these extensions to slow down water flow, and they coat it with mucus.
Oh my.
Okay, I think that's a good hole, thank you.
That's good. All right.
Tubular.
What's that little?
Oh,
guy?
there's
Yeah.
A
someone
worm down
still down there, yeah.
Did
Little
you?
pink thing.
Yeah,
What
that's
is?
probably a mollusk, an aplecophoran.
Did you want it too?
Yes, please. You caught us a very good one yesterday.
Yeah.
Roger that.
Suction back on.
Rog. Thank you.
It's a mollusk without a shell.
Oh.
Anything else you guys want here?
No, I think we can stow it. Thank you. It's
Roger
plenty for us to look at later.
Sure thing. Full wide, please.
While we're here, Jess,
Yeah?
you see that black rock that you broke off and when we landed?
Yep.
Can we take a look at that?
Sure thing.
That was part of a flange of the arm broke that off?
Not sure if it was resting there or if it broke off.
I think it broke
Can we
it
zoom in maybe and just see what it looks like a little closer?
I think it broke off from the ledge when we came down.
All right, Jonathan, go ahead and start pushing in.
Definitely saw it come off of the ledge,
Yeah.
but don't know if it was part of it or resting.
Ah, true.
All right.
I think that looks like a pretty decent rock to me.
Checks a lot of boxes, huh?
And it'll fit.
That is true.
You guys can grab it.
Sure thing. Full wide there, Johnson. Or partial wide is good. That's great. Yeah, it looks like it was part of that crusty bit on top.
Does indeed.
Well, we'll find out. If nothing else, we'll learn that this is not what we're after, but we'll let Kira educate us tonight.
Roger that. Full wide, Jonathan.
Hey, it's Kira from the lounge. I just missed that. Did you guys pull it from the ledge?
We were going to try to break something off the ledge, and then Caitlin noticed that we knocked this piece off at some point. And so we thought that might be a good candidate for you. It's a little thinner than I realized, but
Are you keeping it?
we were planning on it.
Can I look at it one more time?
Yeah, bring it in.
Yeah,
Jonathan, go ahead and push in, please.
Coming in.
and a slow rotate would be awesome.
Sure thing.
Yeah, is there any way to see the edge that's in the inner part of, or on the outer part of the claw? I don't know if that makes sense, towards the fingers.
More like this, you mean?
Yeah. Yeah, exactly.
Looks very sedimentary.
It is, and the fact that it broke pretty easily makes me think it's probably not phosphorite.
Sorry about that.
That's a lot of good biology on it, though.
Zoom.
Yeah, if you guys are interested, I can characterize it for you.
Yeah, I think it's a good rock for Lisa to sample. So
Let's take it.
Take it, Raj. Full wide, please.
Oh.
Could we aim to put this in the starboard bio box, the forward inward box? Inboard box.
Forward, inward. Sure. Sure thing. Anything floating in this box that we need to know about?
Nope, there should be nothing in that box.
Roger that.
Oh no.
Raj.
This is going to be sample NA12443, and it's going in the starboard bio box, number 2, yellow.
This one here?
Mind if I do a zoom on Argus?
Sure thing. Just confirming this is the correct box?
Yes, inboard, forward, yes.
Great.
Does appear there's a rock in the forward left.
Yeah, I think we have a lot of rocks already on us. Earlier team got excited about all the rock samples.
That would be rock 2, I believe, yeah?
Oh, that's actually going to be
Rock
rock
3,
number 4.
Rock number 4.
Anything else around here that you guys are interested in?
I think we can get up and mosey on.
Yeah, just one nice thing would be a really close-up of that beautiful sea cucumber there, right where the lasers are. We haven't really got a good frame grab of that.
Sure thing. Go ahead and fall on in there, please, Jonathan.
Yeah. If you could center it up, that would be great.
Sure thing. Stand by.
So this is a group of sea cucumbers.
Hey.
It's part of the Solidae. It's got its head end away from us with all those feathery tentacles out, and it's filter feeding.
All right, Jonathan, are you full in?
Yep.
And it's permanently stuck on a rock there.
I'm getting some great screen grabs right now.
Right.
I love the color. It's almost like a white, pinkish opalescent.
Yeah.
Okay,
Full
thank
wide,
you very
please.
much.
Sure thing.
Some nice sponge up on top, Paul. Oh, we've already got that sponge.
I'm going to run suction again. I don't know if I saw that little mollusk go through.
Roger that.
I think the yellow one, they sampled just before we came on.
Same as before. Yeah.
Jonathan, full wide on Argus, please. Ooh,
hello, Ray.
Cool. You want to
Very
push in on
cool.
Herc a little?
Oh my goodness. What a special treat.
Wow.
Oh,
Oh,
Can't
I
go
zoom
that
in
way.
on
oh my goodness. We have a visitor.
Uh-oh.
What are you doing?
Oh, don't
What?
take the rock.
That's
Don't take the rock. Oh.
What are you doing, friend?
Curious, I guess.
Blinded.
Come back here.
Huh, interesting.
I wonder if that was purposeful.
Yeah, I don't know.
Just a bit blinded by the lights.
Full wide on both, please.
Blinded by the lights.
knew that was coming.
Had to do it.
All right, Rennie, I'm ready for you.
Hey, that should have
Copy
been a
that.
rock.
Bridge now. .50 meters, 095.
Did you see it go in, or is it still?
I can't tell. I saw something that looked like it, but it's buried now. So
Roger that.
So I just thought of a deep sea joke.
Oh, yeah.
What type of deep sea fish got an A in geometry?
Anglerfish.
Yes.
Ah.
That's pretty good.
A little
I like that
far-fetched,
one.
but
If it was a tropical, would it be a triggerfish?
oh yes.
Ooh.
Triggerfish would be good at trigonometry.
Jonathan, you want to do a partial, please? So these are all rounded carbonate crust. Is this what that
Looks
is?
like it.
Interesting.
Yeah, earlier in the dive, we had these perfectly round ones. It was quite remarkable.
Yeah.
Ooh, another octopus.
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 All right, we have a vote for octopus, Raj.
Yep. The
Carbonate
audience
crust can
demands.
wait.
There it is. We saw one earlier on this dive that looks similar to this one.
Go ahead and start pushing in there, please.
Wow, yeah.
Oh, very cool.
Surrounded by sponges
Yeah.
for a pillow.
Looks like a
Is that a
nice little
sponge behind it? Or that almost
think
looks like a
sponges
solitary
surround it.
Ascidian.
obsidian.
Those
Are you
were,
looking at the
Did you-
yellow or?
the white just
Oh, the white.
above it. Can you pan up just a tiny bit?
Hello,
Sure.
octopus friend.
I think the little yellow ones last cruise they were saying were a type of tunicate.
Oh, a type
that
of
guy,
tunicate.
that looks like a
Oh.
solitary ascidian. That would be a great sample if we could collect that.
Roger that. Full by, please.
Since we have not had an ascidian yet.
Octopus is going to guard it.
Oh, sweet.
Maybe.
Second.
Reg.
It's like a purple thing on lower right next to
Bridge
the
now.
yellow sponge.
All right. Are those the? I think those are
Pole
the feeding
position.
from the Solaris. It's a little purple though.
Yeah.
All right, so trying to disturb our friend here.
Kira's asked for a screen grab of this environment
Got it.
and lots of grabs before and of rock samples too.
Rog.
Thanks,
Go ahead and
Kira.
push on in there, please, Jonathan.
Coming in.
And you see she wants the note about the rock. Looked like it was from a sedimentary outcrop.
Yes, we have recorded that note,
Awesome.
Kira. So those yellow would be really cool too. Solitary tunicates.
All right, full wide there, please, Jonathan. Try not to disturb our sleeping friend there.
What do you think, Greg? Is this a slurp or a slurp grab for that ascidian?
I think it'll come off easily enough with the slurp, unlike those tube worms.
Yeah,
And,
I think it's a pretty good sample for us.
yeah, coarse chamber, probably a good call.
Yeah, we've got slurp jar number 6 open.
There's the colonial sea squirts also where the octopus is. Maybe not enough volume for you.
Yeah, those will be a challenge.
Do a quick flush.
Yeah, go ahead.
So ascidians look very primitive, Greg, but they're among the most advanced of the invertebrates, if I recall correctly. Hemichordates or chordata.
When you zero it, I'm going to re-get my grip.
Yeah,
Zero.
Reg.
if you think of our evolutionary branch with the fishes and ourselves, the next closest relative is these sea squirts, which is pretty hard to believe when you look at them. But when you look at their babies, they have a little phase called a tadpole larva.
Really?
It's very reminiscent of a tadpole, really. It does have in it a tail with a cord that's
Yeah, look over
similar
at
to our
at the octopus with yeah,
spinal
so don't
cord.
hurt him.
It looks like they
Okay, Jonathan,
took
push
an
on in there,
evolutionary
please.
branch to become filter feeders sitting on the
Right.
bottom,
Little wide.
and the other evolutionary branch led to retaining
more.
that cord and becoming
great,
the
thank you.
vertebrates.
It's quite remarkable.
Fascinating.
Jar number 6.
Yes,
correct?
jar number 6.
Okay.
This is going to be, oh, we'll wait for the sample.
All right, Jake, go ahead and come on. Maybe 70% is good.
It's 50%.
Roger that.
So this is
Even
what's
better.
called a solitary ascidian or sea squirt. It has two openings. One brings water in, and then there's a basket to filter out the food, which then goes down little grooves to the mouth, and then the now clean water goes through the basket and out another hole called the exhalant siphon. And meanwhile, down at the base is
Sorry.
a complete gut.
All
And these are hermaphrodites. They're
Right.
male and female at the
Beautiful.
same time.
Do you want to slurp a little more around here?
Yeah,
Because
since we can.
Yeah.
Yeah. Might disturb the octopus, but it'll move out of the way, and then maybe we can tell what it is.
See it's coming through.
You could maybe take the orange, yellow, little yellow encrusting
Ooh, there it is.
Got it.
There it
Nice
is.
job.
sea squirt.
Nice.
This is going to be sample NA124, 104, and slurp jar number 6.
Yeah, I just want them to grab onto these because they have a tendency to be a little grabby when it comes to our slurp.
Oh, the octopus might grab you?
Yeah, we've had that in the past happen when we go too close to them.
Okay.
Up to you
Yeah,
guys.
we don't want to suck it up, that's for sure.
But you want back here, kind of a dusty
Yeah,
rock?
you can go away from it a bit. Yeah.
Roger that.
Can you get to the tuna crab up? A
Yeah,
little
sure.
bit further? Yeah.
Go, Jake.
50%.
Rog.
Oh,
darted.
You son of a gun.
He's wise to you.
Oh.
Oh, yep.
Yeah, Jake. Yeah,
Full
get him with
jam.
the extra
Full
suction.
jam.
Yeah. Good thinking. Well, we got 2 of them in there now.
2 of them, yeah. See if they've come through. Ooh,
Dust
cloud.
storm.
must be in
I
there.
think it's a tuna crab. Anyway, we'll do some genetics on it and figure out what it is.
So I'm staring at this white sponge, Caitlin, and do you think
There
that's
is,
the one
there
they
is.
collected this morning?
You got 2 in there, though.
Oh, there's 2.
Oh, no,
Okay.
not yet. There's going
Yeah,
to
waiting on the second one.
Yeah.
Have an idea what that purple thing is just on the full right side of the frame? A little purple pebble.
Right to the right of the
Oh,
yellow.
I think it's a sponge.
All
Is that
the
one of
way.
those moldy cheese sponges,
Mm.
or is this not
Ooh,
there?
like very to the right.
Yeah.
The little
that
Death
purple
Star sponge on the bottom there.
looks like a little
Oh,
speckled.
it's the moldy cheese? Is that?
Yeah.
Like,
That's what I, well, I don't know if that's what it is, but that's what it looked like.
moldy cheese sponge? Yeah.
Yeah.
I like
It looks like there might be more of that yellow. I'm not sure if it's the same type or not by the moldy cheese sponge. Or the yellow sponge over there. Anything else you guys want over here?
Maybe down to the lower right where there is that yellow and the black and white, or black and gray,
Sure thing.
if you can
Come a
reach
little
there.
wide, please.
Got it?
Right, there's some more of it right at the bottom of the screen.
Oh, yeah. Nice. Nice eye.
I think it is a sponge. But,
Can you get that rock out of the way? We can't see.
I know, right? I would.
Put that there.
I would gladly love to play Frisbee underwater. All right, I'm going to reset myself up here.
Okay, Kara. We won't make fun of your rock anymore.
All right, Jake, go ahead.
Same slurp jar, right?
yes, please.
Yeah.
Yes.
Push it in there, please, Bit Jonathan.
Coming in.
That's great. You want all 3?
Yes, please.
Roger that. Oh, it's on a big rock. Let's start with this guy. Oh, you're kind of big.
Got him.